good morning, students. I hope I'm audible.
Good morning, ma'am.
Morning. Sorry to wake up you early. So we'll continue last lecture that was not done yet. So I'll just quickly go through what we have done earlier. Yesterday's lecture. So yesterday we discussed about pathogenesis. We discussed about what is disease cycle, right? We discussed how pathogen, what are the different steps in pathogen's life cycle or a disease cycle, right? How they go. So what are primary disease cycles or a monocyclic diseases? What are secondary disease cycles or polycyclic diseases? And we also discussed about the inoculum, right? So what is primary inoculum? What is secondary inoculum? And how primary inoculum it causes primary infection, and how secondary inoculum can lead to secondary infections, right? And then different sources of inoculum. And we also discussed about the host range of the pathogen. That I told you this yesterday also, that not all plants are the host of any particular pathogen. So pathogens are selective in their plants which they infect, and so definitely not, they are not going to infect all the plants. So reason could be many. Plants could be resistant to that particular pathogen, or they are not susceptible to that particular pathogens, the other way around. So there could be many reasons for that. So it's that every pathogen has its host range, right? Then we discussed a bit of a disease triangle also, though the disease triangle we are going to study in our next module, which is basically about epidemiology and a few other concepts also, right? And we discussed how they are being dispersed, how pathogens they are distributed and disseminated, and difference between distribution and dissemination, where we said that distribution is when pathogen is spread across within the same area, within the same geographical area where they were once established. Whereas dissemination is the term used when that particular pathogen is somewhere it is being introduced into some new area where it was not present earlier. Then we also discussed about inoculation and inoculation potential, what is the incubation period, and in detail we discussed about the penetration, right? So penetration process is divided into three parts: pre-penetration, penetration, and post-penetration, and what are the different changes that takes place during this process. And then the spore germination, how the different fungal spores or bacterial spores they are being germinated. So the fungal spores, we know there are certain structures such as a germ tube is formed, and it also form aspergillus or infection hyphae or hostoria, right? So different kind of a structures it forms to get entry into a particular pathogen. So all these things we discussed yesterday, and today we are going to discuss start with the infection process, right? So infection process is basically how how a kind of a relationships of the pathogen with the susceptible cells of the host, how it is making that relationship with the uh, cells of the host. So that process is called as an infection process. And there are, see, we define this timeline of infection or this process as into two parts. So one is basically outside the host, and then another is a inside the host, right? So there are different steps which takes place when pathogen is outside the host. What are the different steps that it takes place to gain entry? What are the different modifications, right? And what happens when once it is, or pathogen it gains entry inside the post, right? So outside the post, this we have already discussed that spores, they will germinate, they will form a germ tube, right? And then there will be a prosporium formation, or a penetration peg, right? So these steps they are taking place outside the host. And certain sometimes what happens, the plants they also form certain defenses, right? Certain defenses, or there are certain structures in the plants which helps to avoid the entry of the pathogens. Could be the corky layer, could be the cuticle or a waxy layer outside the plant leaves, which again to some extent they inhibit or they just block the way of the pathogen, right? So all those things can happen. Cork and a legionella layer also happens, right? And once it gains entry inside the pathogen, then number of things happen here also, right? Could be some pathogens or fungal pathogens, they could release certain kind of toxins also, and then so that they can get entry into the plant trend, right? So once they get entry, then there will be a reproduction also. Then plants also, they have a certain defense mechanism, right? The systemic acquired resistance, or sometime they can hypersensitive action can lead to the death of the few cells where the pathogen has tried to enter, right? So certain kind of steps are being taken by the pathogen also and the plant also in defense to that particular pathogen, so that they can stop the entry of the pathogen inside, right? So there are certain constitutive defense mechanism, could be the chemicals or could be the structures out which are present in the plant, which would help in avoiding the pathogen, or at least they will try to avoid the pathogen entry into the plant, right? So successful infection, how do we know that infection is successful? It's definitely symptoms, right? So once we see the symptoms on the plants, so definitely that shows that the infection was successful or is successful. Again, then symptoms are again a local symptom or a systematic symptom. So systemic symptoms. So local means it is just in that particular area. It has not spread to the other part of the plant. Systematic means it has spread to the other part of the plants also. And sometimes infection is a latent. So when we use the term latent, means it is there present in the plant, but still it is not showing that much evident symptoms. So infection is a latent, not visible symptoms, right? So invasion and reproduction are two stages that occur during infection as pathogen invade different cells. So that means it is getting entry into the different cells in the plants, and it is growing also, and it is multiplying also. That means growth and reproduction of the pathogen inside the host plant, right? Then infection process is again affected by various factors. Here, then resistance and susceptibility of the host. So this is also important, right? Sometimes plants have certain R genes or resistant genes. So due to the presence of those genes, the pathogens, they are resistant to particular pathogen, right? And if it is susceptible to, so definitely if it is susceptible to the pathogen, it is going to show a lot of symptoms and a lot of changes. If it is resistant to the pathogens, then definitely the pathogen cannot multiply in the plant further, right? Then aggressiveness and virulence of the pathogen. How aggressive is the pathogen? How virulent is the pathogen? How much infection it can cause in a particular plant, right? And then there are environmental factor, host nutrition and the pH, and the incubation period. Incubation period, again, as I told you, is the time when the pathogen first penetrate the host or enter the host till the time it shows the first symptom. And incubation period can also, yeah.
आपके group call कभी
Hello. Yeah, Nandini, are you saying something? Yeah, hello, Nandini. Was someone saying something? I just heard something, right? Yes, Nandini, are you saying? Okay, fine. So, incubation period is the time when the pathogen first enter the host till the time first symptom is prepared. So that many days or that many hours are called as the incubation period, and it differs from different pathogens and different like viruses could be different, and even the viruses also, the group of different viruses can have a different incubation period, and similar is for the fungi also, right? Invasions means pathogens, they enter to variable extent in the host. So it is invading a host. So how much it has invaded a host, whether it has gone to the whole host or a root system only, or it is invading the only shoots of the particular plant only, or the even the fruits also. So how much it has invaded a particular host, right? The different pathogen, they can invade their host as an ectoparasite or an endoparasite. So ectoparasites are usually outside the host. That means they will not penetrate very deep. So only within some epidermal layer or through that layer they can just multiply and they can produce their spores, such as powdery mildew. Then endoparasites are, that means they are going inside the plant systemically. That means they are going inside, they're moving inside the plant, maybe through the xylem or a phloem or through any other part, such as wilts and viruses. They are endoparasites, right? Then subcuticular pathogens, that means they are affecting somewhere near the cuticle only, not beyond that. So symptoms such as apple scab, which is caused by Venturi inequalis, they show the symptoms on the cuticle. Then subepidermal, that means they will penetrate through the epidermis to that part, such as wheat rust are the example of subepidermal pathogens. And then we have a vascular pathogen. Vascular pathogens are mostly wilt causing pathogens which go and invade somewhere near the xylem and the phloem of the plants, right? Vascular tissues. So they cause the wilting or other similar kind of diseases. Then ectoendoparasites like potato canker. So they are causing symptoms on the outside the plant also and inside the plant also. So those are called ectoendoparasites, right? So invasion behavior, as I told you, ectoparasites example is the powdery mildew. And we have a subcuticular apple scab. You can see the mycelium near the cuticle, subcuticular, right? Then we have a subepidermal in the wheat rust and endoparasites such as bacteria or viruses inside the plant. Then growth and reproduction. So how they are growing and they are reproducing, and then finally they are taking exit from the plant because they need to infect some other plants also, right? So different pathogens, they grow intercellularly and intracellularly inside their host. Growth and reproduction rate depend upon the invasion behavior of the pathogen, and then the method of the reproduction, how it is asexually or it is going under through a sexual reproduction, right? Then how it is replicating some, for example, some viral pathogens, how they are replicating, some microscopic pathogens they replicate, right? And growth and reproduction is affected again by the pathogen aggressiveness, how aggressive is the pathogen, how susceptible is the plant tissue, and what are the environmental factors inside the host. Environmental factors definitely outside the plant, how it is, in which environment it is there, but still the factors responsible are also inside the host because pathogen has also gained entry inside the host. Now, what are the factors there which might affect the pathogen growth, right? So to general form of a reproduction, we know they reproduce through the spores, sporangiums or zoospores, which are formed by, this is in case of a fungus. These are formed by a spore-bearing branches which are called sporangiophores, and they form sporangiospores or zoospores. For example, example is a Phytophthora here, the sporangia and zoospores of a Phytophthora, and then sporangia of a downy mildew. So different spore, fungus produces different kind of a spores, and they reproduce by different means. So on that basis, it is being divided. Then they can form a conidias also. They are asexual spores. They are non-motile fungal spores, and they developed externally or they are liberated from the cells where it is being formed, right? So they are asexual spores, conidia. Example is a conidia of a powdery mildew, then conidia of a helminthosporium. These are the example. They form conidias here. Then chlamydospores. So what happens sometimes, the hyphal cells, they directly lead to the formation of a certain spores which are called chlamydospores also, right? So here example is again Phytophthora. So chlamydospores formation is being seen in the Phytophthora, the example here, right? Then we have a sexual spores which may occur in the mycelium, right? They are enclosed in a fruiting body. They sexual stage is usually once a year, not throughout the year. Only once a year they form these kind of spores. Examples is the apothecium, which is being produced by sclerotium. So it is the fruiting body on which the spores are being produced, or Clistothecia in a powdery mildew, which produces a spores. This is again a perithecium in Venturi inequalis. So again, this is you can see here the flask shape, and this is a 40x in a compound microscope. So in a 40x compound microscope, this Clistothecium or a perithecium or any kind of a fruiting body can be easily seen. We don't need a high resolution microscope. In a compound microscope only, in the 40x, these things can be clearly seen. So we can see it is a fruiting body is there in which there we can see the spores inside that also, right? So these structures can be clearly seen when we cut the infected tissue and just try to see it under the microscope. Then factors influencing, this is again same, resistance and susceptibility of the host. If the host is resistant, then definitely pathogen cannot multiply to a larger extent, right? Then predisposition factor. This I discussed yesterday also, that some factors made plants vulnerable to few pathogen attacks. Could be the environmental factor, adverse environmental factors, wounds, or any other thing, right? So due to those factors, if they are affecting plant, these pathogen become more susceptible to these plants become more susceptible to the pathogen, right? So plant vigor, which is determined by the age and the nutrition. So malnutrited plants, they get more infected. Young plants, they get more affected by the pathogen as compared to the old plants, right? Then the temperature, humidity, light, nutrition, and other chemicals. So these are the factors which influence the infection also. Then wounds caused by microbes or a mechanical means. So wounds are the very easiest way of entry into the host by different pathogens. It could be the fungal pathogens, it could be the bacterial pathogens, it could be the virus pathogens, or a nematode. So wounds are the easiest. If any plant is wounded, they become accessible to different kind of a pathogens, right? Or a plant exudates, that means the secretions which are being they are being done by the plant, or there are certain secretions released by the plant. So through that also they can get entry. Then certain cultural practices. So cultural practices means all the practices that we follow in the field, such as thinning of the plant, or we are removing the weeds from the plant. So if by that means we can somehow touch the plant, different plants from one plant or another plant, infected plant, we are touching infected plants, and then we are touching the healthy plants also, so we can transmit the pathogen, right? Then aggressiveness of the pathogen and certain other environmental factors. Then types, effect of infection. So what effect the infection causes on the plant? It could be the structural changes on the plant, or it can lead to physiological changes in the plant, right? It could lead to the tissue disintegrations also. It could affect the growth of the host also. It could affect the auxin, gibberellin, and cytokinin, different kind of hormones also. And it also affect on uptake and translocation of water and nutrients. So definitely the movement of the nutrient in the plant is also affected, could be affected by the pathogen. It also affect on absorption of water by the root. So different plant. Just a moment. So different functions of the plant can be affected by the pathogen, and it totally depends on the pathogen. That if for example, we know if pathogen is in xylem and phloem, then definitely it is affecting the absorption of the movement of the water in the plant, right? It could lead to the wilting. So it also depends upon the pathogen, that in which way pathogen has accessed the plant, or it has got entry into the plant, so where it is residing presently. So this way sometimes they affect the chlorophyll production in the plant, so that leads to the yellowing of the leaves also, right? So different ways they affect plant. Effect on the translocation of the water by the roots, right? So as I told you, fungus or mycelium spores or bacteria, they are present in the xylem, right? Or the enzymes, they could lead to the different kind of the effect there. So movement of the different nutrition and the movement of the water through those xylem and the phloem of the plant, right? It can affect the transpiration also, right? The physiological and pathological wilting, right? So the term, see, you see here we are using two terms. There's a physiological wilting and there's a pathological wilting, right? So pathological wilting is being caused due to the pathogen usually, and physiological wilting is caused usually due to the stress in a water, excess of a water or a less of a water condition. So two types of wilting are there, right? Then effect on translocation of the nutrients and their deficiency in the plant. So it could affect that way also, right? This is just the composition of a structure and composition of a cuticle, cell wall, or a folial epidermal cells, and how This is just an example to show that how there's a how internal structure of a cuticle of a wall or a folio epidermal cells looks like, and then pathogens, they can affect the cuticle or epidermal wall or other plant cells in a different way. Then plant cell wall composition, this we all know that it has a different composition. It could be cuticular wax could be there. Then cuticle, again, it is formed of derivatives of hydroxy fatty acids or dicarboxylic acid molecules. Then we have a cellulose also, right? So there are number of different compounds of which the cell wall is made up of, right? Then hemicellulose, we also know, again, it's a branch polysaccharide which is composed of the hexosyl residues, right? Different components of a cell wall, they have a different composition, right? So okay, compositions, I guess we'll not go in detail. Then enzymes, they play a role in a pathogenesis, different enzymes, right? So mostly play a role in a soft rot diseases which are caused by fungi and bacteria. And these are also produced by the nematodes, right? Some viral infections are also, we found the release of certain kind of enzymes. So plant pathogenic enzymes, they disintegrate the structural component of the host cell. They affect protoplast and interfere with the normal function. Usually, enzymes are helpful in disintegrating the cell walls or a cuticle to get entry into the plant cells, right? Then cuticular wax. We know some plants, there is a waxy layer also outside the cuticle also, right? So they are consist of alcohols or ketones or esters or certain hydrocarbons, right? So these are found as a layer or projections on the from the cuticle. Above the cuticle, we see a single layer, right? So mostly penetration by mechanical forces of the pathogen, fungi, and the parasitic plant. It can prevent to certain extent if it is there, right? Some pathogens, they are found to produce enzyme which cause degradation of this cuticular wax. For example, Vaccinia hordeii and Pastolotia. So these fungal pathogens, they produce certain enzyme which can degrade the cuticular wax so that they can get entry into the plants, right? Then we have a cell wall degrading enzymes also, right? So cuticular enzymes, cutinases, these are the esterases that break the ester linkages between the cutin molecules, right? So these degrade the cutin. So they are helped. For example, these are produced by Penicillium, then Eris also, then Streptomyces also, Botrytis cinerea also. So these enzymes are being produced by different fungal and bacterial pathogens. Okay, so this is how cutin is degraded. Cutin is a polymer. So with the enzyme cutinase, the fatty acid derivative, it initially small amount of enzyme is produced, and after formation of monomers and oligomers, it induces more cutinase production, right? Then these enzymes, what they do, they soften the cuticle. They make it soft. Some of the fatty acids which are present in wax also induces cutinase production. Then glucose suppresses the expression of a cutinase gene. So this is how a cutin is being degraded. Then we have a pectin degrading enzymes also. Again, pectin act as a cementing material between the cells, right? So enzymes such as pectinases or pectolytic enzymes, these are produced by fungi, bacteria, and nematodes. And then enzymes produced are pectin esterase and then polygalacturonase or pectin transaminase. So these are the different enzymes, pectin degrading enzymes, which are being produced by the pathogens. Okay, so we'll not go into very detail of these enzymes, right? Then we have a polygalacturonases also. These are called chain splitting enzymes, right? So again, they are of many types such as polygalacturonases, polymethylgalacturonases, right? So this polygalacturonases, they act when a substrate is a pactic acid, whereas polymethylgalacturonases act when substrate is a pectin, right? So different on a different kind of substrate, different kind of enzymes they act. Then pectin transaminase or pectate lysase. Again, pectin is also broken down by these enzymes. So they eliminate water molecules rather than hydrolysis, right? So these are produced by Erwinia carotivora, which cause soft rot of a carrot. So soft rot of a carrot means they cause the softening of a tissues or a fruit tissues, basically, right? Rotting of the So these pectolytic enzymes, they cause liquefaction of the pactic substances, which hold the cell together and lead to maceration of the tissue. So when tissues are soft or they are macerated, it is easy for a fungal or any pathogen to move and entry to that particular host, right? Then cellulose degrading enzyme are cellulases. These are again, they help in cell wall digestion is being done by these enzymes, such as in fungi such as Rhizoctonia or Fusarium or Paccinia graminis criticae. So these enzymes are being produced. Then hemicelluloses, right? So these degrade various complex polysaccharide polymers and which are found within the plant cell wall and between the cells, right? For example, xylanases, manases, and others. These are being produced by Sclerotinia sclerotium. Then lignin degrading enzymes. So certain plants, they have a lignin as a composition, right? So these enzymes are produced by basidiomycetes and which cause wood rotting of the forest trees mostly. See, to get entry into plants or vegetable crops is still easy. To get entry into the forest trees or a fruit trees, it is difficult, right? Because their composition is such, the bark is very hard, so it is very difficult for a pathogen to get entry, especially fungal or a bacterial pathogens, right? So for that, they definitely need to produce a different kind of enzyme which can degrade their cell walls or which can make their entry possible through that particular tree. Then protein degrading enzyme, proteinases, then starch, we have a starch degrading enzyme, then we have a lipid degrading enzyme, right? So we have a different kind of enzymes. Then role of toxins in plant pathogenesis. So toxins can be a pathogen derived molecules which are produced by the pathogen. So could be a toxins, could be a polysaccharides. They can produce certain growth regulators also, or enzymes also, which can affect the plant and their movement make easy in the plant, right? So toxins can be exotoxins or endotoxins, right? So toxins in a very low concentrations are toxic to the cells of the susceptible, right? Host. So in plant pathology, toxins are the product of pathogen in host or a host pathogen. Either they are produced as a result of infection in the host, or it could be produced as a result of a host pathogen interaction in a particular plant, right? So could be if mycotoxins are the toxin or a toxic substances which are being produced by fungi, which are not or we can say which are harmful to the animals and the humans also. Then mycotoxicosis, again, disease of animal and humans caused by consumption of a feed and a food which is invaded by toxin producing fungi. So these toxins are actually toxic to both humans also and a plant. So sometimes what happens, the certain diseases in the plants are caused by the fungus and they produce toxins, and if those plants are consumed by the human, it can cause diseases in the human too. So this is basically due to the toxins which are being produced, right? So if we compare between toxins, antibiotics, and enzymes, so toxins are low molecular weight, even the antibiotics are low molecular weight, whereas enzymes are high molecular weight compounds. Toxins are toxic to the host, whereas antibiotics which are being generated are toxic to the microbes, right? We are using antibiotics against the pathogens, and enzymes, we know they act on a particular substrate, right? Toxins, they have a killing factor, whereas antibiotics have an antagonistic factor, and enzymes, what they do, they make the tissue soft so that pathogen can get entry into the host, right? Macerating factor, softening of the tissue. Then there are certain toxins called phytotoxins. So substances for which, phytotoxins, we can say these are also products of a parasite which induce a part or some symptoms which are caused by the pathogen, right? So non-specific and a non-relationship between symptoms, virulence, and amount of the toxin produced. So phytotoxins can sometimes be produced by the parasites. Phyto means plant toxins, right? In the plants when they are affecting plants, such as alternaric acid, which is being produced by Alternaria solani, where it infect a particular plant. Then we have a pathotoxins also. Again, these toxin will produce all kind of symptoms of the disease. And again, this toxin which is being produced by the pathogen will be directly related to its ability to cause the disease. So more the toxin is being produced, that means more it is causing a particular disease in a particular plant. For example, Victorin is a toxin which is being produced by Helminthosporium. So this is example of one of the pathotoxins. Then we have a vivotoxins. Vivotoxins is again a substance which is produced in infected host by the pathogen, or its hosts which function in the production of the disease, but it is not initiating the disease. It can be produced in a particular plant as a result of the disease, but itself it is not initiating the disease. It is not a factor in initiating a particular disease. Then they say there are 4 criteria which are suggested for vivotoxins. It is a reproducible separation of a toxin from a sick plant. We can isolate it, we can purify it, or we can study its chemical characteristics. Then induction of at least a portion of the disease syndrome by placing toxin in a plant. So if we place only a particular toxin which is being isolated on a plant, some kind of a symptoms can be regenerated, right? Then vivotoxins are non-specific, so they are not particularly produced by a particular pathogens only, right? Fusaric acid or pericularins or lycomerimycin. So these are the example of certain vivotoxins. Then ecological roles of toxins. They can cause a phytotoxicity. They can be pathotoxins also, right? If they can alter plant gene expressions also, right? So prevent defense. So that plants, when certain pathogen they affect plant generate certain defense responses, right? So these toxins can prevent these defense responses of the plant. So question mark यहाँ पे इसलिए है कि this is still under not very clear. They say it could happen, but it is still not very clear, and it can contribute to pathogens fitness also. It can alternate the plant metabolism also, right? So again, it can play a dual role with virulence or pathogenicity determinants. These toxins can play these kind of a role also. Then possible role in the diseases, the necrotrophic fungi, for example, they causes necrosis and cell death, and it also suppress this defense response of the plant. Then in some certain other hemibiotrophic fungi, they prevent cell death and they suppress the defense response, right? So biotrophic fungi, again, known pathotoxins, they are not generally associated with only biotrophic fungi, right? However, this does not indicate lack of such toxins, since these are less traceable systems for experimentation. So they might be present there, but yes, they are less traceable there, right? So they could suppress resistance. It is still a question mark, or they could be elicitors of a race-specific resistance. They could do that, right? For example, some rust resistance genes such as PS2 at toxins, sensitivity gene locus vivae, they have been identified. Also, mutagenesis result suggests that these are the same genes. So there are some studies which are being conducted which suggest that these rust resistance genes are affected by these kind of the toxins. Then toxin characteristics, they can be host selective, worse they can be non-selective or non-specific also. They can cause a programmed cell death also, or they can prevent cell death or a hypersensitive response also, such as HC toxins. Hypersensitive response is usually by the host, right? And hypersensitive response by the host is in its defense mechanism. So basically, pathogens, they don't want hypersensitive response, because hypersensitive response kills the cells, kills their own cells. So pathogens, they are not able to multiply in the dead cells, right? So they want to stop that hypersensitive response, and this can be done by the toxins also, right? So these are the few characteristics. Then chemical classes could be, they could be aflatoxins, they could be T toxins or circotoxins, polyketides. Then they are terpenoids also, for example, phenyltransferases in biosynthesis, fusicocaine, or we have a lolitrum, and we have a gibberellic acid. So there are different classes also chemically, right? We'll not go through this. Mode of actions, they can change in a cell permeability. For example, fusaric acid, Victorin, etc. So they change in a cell permeability. So that will help pathogen to get entry. Again, they also disrupt normal metabolic processes, right? So inhibition of polyphenol oxidase by pericularin, then formation of a chelate ring complexes by a heavy metal. So they normal metabolic processes in the plants are disrupted by the toxins also. Again, in addition to toxins, the necrotrophic pathogens, they produce phytotoxin proteins also, right? For example, the Fusarium oxysporum secreted necrosis and ethyl-inducing protein causes plant cell death, and NEP-like family in fungal proteins की characteristic है. So they produce certain proteins also, right? So they can function by interfering with integrated of the plasma membrane. So they can disrupt the plasma membrane, right? So the disruption of cellular integrity is then detected by host plants, leading to activation of the defenses also, right? So in contrast to phytotoxins, NLPs are unique in their conserved toxicity to dicot plants only, not in a monocot plant, right? So these are some of the research which has been done by the people, and they have said that there are certain necrosis inducing proteins are also being identified, right? Then we have a dissemination of the pathogen. So that means how they are disseminated. So how inoculum can be transferred or transmitted, like passive through wind, water, insect, man, animals, or a machinery, right? These are the passive ways of a dissemination. Then active could be the on move on their own. For example, bacteria or zoospores or a fungal spores, if they are taken to a longer distance. So that means that is the active mechanism of their dissemination, right? How does this pathogen survive? So they survive in the infected crop debris, they survive in the seed, they can survive in the soil, they can survive on the growing plants, they can survive on the infected material on the host plants, then in the propagative material, they can survive in an alternate host, they can survive in a collateral host, or they can survive as the dormant structure. So when we talk about the alternate host and the collateral host, these are two hosts, but they are different, right? Alternate host means a plant, a host plant which belongs to the same family, right? So host plant which belong to the same family on which these can survive. Collateral host means a plant which belongs to the different family or a different group. So if they survive on that particular host, it is called as a collateral host of that particular plant, right? So this is basically all about the infection process, how infection is taking place, how pathogen is getting entry into a particular plant, how it is multiplying there, what are different kind of the toxins or enzymes being produced, what are the structural defenses of the plant, right? How, what are various structures, such as lignin or wax layers or corky layer. So how they prevent these are called structural defenses of the plant, right? How structurally they can defend themselves against the particular pathogen, right? So just go through this. I have also uploaded the PDF file also, right? So just try to starting to the end, just try to go through this and just try to correlate the events which are happening. And this is in very short. I'm not explaining anything in very detail because that will take again, that is not, this is supposed to the basic plant pathology, as an introductory plant pathology. So we are not supposed to discuss in detail everything. Again, that require 2-3 more courses, right? So just try to go through whatever we have discussed today and just try to read the PDF files also and focus on the terms, right? Focus on different terms because different terms, they have a different meanings. There are a lot of definitions. There are a lot of, definitions which are going to come in the exams also. So you need to focus on the terms also, right? Alternate host, collateral host, like I said there. So all those terms you should know. Inoculum potential, क्या होता है, right? Aggressiveness of the pathogen, क्या है? Virulence of the pathogen, क्या है? So all those things you need to focus on that, and you need to know that what does that, all those terms, right? So I guess we'll stop it here today, and then we'll maybe tomorrow we'll come up with the next topic again in this particular subject only, or in this line of progression of the infection only. We are going to discuss some other topics also, but still just try to go through this, right? Because once you'll read them again, then you'll know whether you have understood it or not. So just go through this lecture again, lecture yesterday also that we have discussed, and today also, just both of them, they are related. And then tomorrow we can take the discussion, or we can take the questions also, if you have any questions or if there is anything that you have not understood, right? So we'll stop it here today, and we'll meet, I guess, let's meet at 11:20 for the next class, right? So there will be 7 minutes break, and I'll take attendance in the next class only, right? So I'll stop here.
Thank you, ma'am.